Good day. I'm Joseph Noor. I'm a medical doctor and a biochemist, and I'm the author of Document X, Direct Evidence of God's Existence. I often get people protesting that evolution is just a theory, speculative science. But in science, a theory means something different. It's an idea that is based on well-founded principles supported by a mass of data. I think it's important just to distinguish between an hypothesis and a theory and fact. An hypothesis basically is an assumption made as a basis for reasoning. And then there's what we call a theory. In science, a theory means something completely different. It's something that's been tested over and over again for years. And there's no data that seem to challenge this theory. Einstein's theory of relativity, it's been around with us for more than a century. All the experiments done so far are consistent with the theory. And so the same with evolution. Evolution is considered a theory, but as I said, based on principles and a large body of evidence. And so far, as I said, any sane scientist does not reject evolution because the evidence is just simply overwhelming and indisputable. Evolution is no longer a theory as is commonly misunderstood, but it is a fact. That evolution happened is no longer in question. There is some controversy as to how it happened, such as what were the underlying mechanisms that drove the evolutionary process. A theory or an idea which is being hotly debated and discussed, and there's progress being made, are definite signs of a thriving theory, not a theory in crisis. Now we come to the most controversial aspect of evolution, our cousinship with the chimpanzee. Now, most people I know find this idea distasteful or even abhorrent. But let's put our emotions aside and concentrate on the facts. Remember, the Quran demands evidence for our assertions. Now, we're going to concentrate on the past 7 million years, because herein lies most of the hostility and misunderstanding of the evolutionary process. Fortunately, our knowledge of this time period is reliable and accurate because of sufficient data. There's overwhelming evidence supporting evolution. We touch on embryology, fossil studies, and we're going to elaborate as we carry on. But I just want to mention something, the idea of poor design. Our creationists have never addressed this issue. For example, the spinal cord is not optimally designed for an upright posture. So either you accept the idea of an untidy designer, or that the spinal cord is a work in progress, that is in a process of being refined and improved upon. Remember the word Rab we spoke about earlier, which states that things emerge in an imperfect, crude form, and as time goes by, there's refinement taking place, and the creature becomes better adapted. Now let's resume our discussion here. To provide the conclusive evidence that we share a common ancestor with the chimpanzee, we have to revisit the DNA molecule. Now remember, DNA is a double-stranded molecule and consists of repeated molecular units called bases. Now a human DNA molecule consists of about 3.5 billion bases. Now there are 4 types of bases, and they are labeled guanine, cytosine, adenine, and thymine, and they are abbreviated G, C, T, A. And A on the one strand always combined with T on the other strand, and G on the one strand always combined with C on the other strand. Now, the order of these bases, in other words, their sequential arrangement, determines the type of DNA molecule, whether it's plant DNA, animal DNA, your DNA, or my DNA. Now here is a schematic representation of a segment of the DNA molecule. Note that if you look at this sketch here, G always binds with C and T always with A. Now in this diagram here, you find that gene A is situated next to gene B. A specific group of bases, a large number of them, makes up a gene. And a large number of genes, more than 20,000 of them, constitutes a DNA molecule. Now the DNA is contained in thread-like structures called chromosomes. Now chromosomes are basically DNA stabilized by proteins Here's an example of a typical chromosome. And the ends of the chromosome is called the telomeres. Now, telomeres consist of a sequence of bases which are highly specific. And then there's a narrow segment called the centromere, and its basis too is very specific. In other words, the sequence of its bases. And then you see here the coil DNA and the proteins. The chimp chromosomes are highly similar to human chromosomes. What is more striking, we share about 98% of our DNA with chimps. How did this 2% difference come about? Remember, in our previous discussion, we said that chimps and humans emerged from the same common ancestor. Now, soon after the split, these ancestors would have had almost identical DNA. But as they diverged and evolved independently as a result of mutational buildup and changes, it eventually gave rise to this 2% difference. So if we live maybe another 7 million years, this difference might inflate to 4%. Now, for each human chromosome, there's a corresponding chimp chromosome. What I mean by this is that if you take a certain chromosome, a human chromosome, if you zoom in on a particular site on a certain chromosome, and you find a specific sequence, say gene A is adjacent to gene B, which is next to gene C, which is next to gene D and E, and so forth, you're going to find exactly the same gene sequence at the same site on a corresponding chimp chromosome. Now, as I said, for each human chromosome, there's a corresponding chimp chromosome, but there's one notable exception, and that is human chromosome number 2, for which there are 2 chimp chromosomes labeled 2A and 2B. This explains why chimpanzees have 24 chromosomes and humans have 23 chromosomes. Now, creationists have seized upon this idea and claims that this disproves ancestry, but it actually proves ancestry. Now, what is the compelling explanation for this? Human chromosome number 2 can be perfectly aligned with chimp chromosomes 2A and 2B. Now, what could be the reason for this curious alignment of these chromosomes? If chimps and humans share a common ancestor, then human chromosome number 2 might have formed as a result of fusion of 2 ancestral chromosomes after the lineage of the chimp lineage split from the human lineage. And this fused chromosome in the human line today is known as human chromosome number 2. And the corresponding chimp chromosomes, which remain separate, is labeled 2A and 2B. So what is the evidence for this fusion? Firstly, if you look at the length of human chromosome number 2, you'll find that it's almost twice as long as either chimp chromosome 2A and 2B. Now, the second reason is that we need a bit of explanation here. Remember I said earlier that the ends of the chromosomes is called telomeres. Each telomere consists of a 6 base pair sequence, which is repeated about 50 to 100 times. I've drawn here just 3 base pair sequences, just for convenience sake, obviously. Here's the 6 base pair, each time being repeated. And I've boxed it to facilitate understanding. There's a 6 base pair sequence in each box, and it's being repeated over and over again. And you can see that T always binds with A and G always with C. Now, the picture in reality is actually much complicated. So, moreover, you find that these are not just letters. These are the words G, C, and T are actually complicated chemical compounds. So, the point I want to make is that genetics is a very complex subject. It is specific, but it provides powerful evidence of our ancestry. What they've done then, they've sequenced the human chromosome number 2 and chimp chromosomes 2A and 2B. When I said sequence, they determined the order of the bases. And what they found is that the middle section of chromosome number 2 is very similar to the telomeres found in the head of chimp chromosome 2A and 2B. So what does this mean? With this observation, scientists theorized at some point in human history, the tips of the telomeres of 2 ancestral human chromosomes broke off and they became sticky. And they fused at the sticky ends and formed this fused human chromosome, which we call today human chromosome number 2. And remember, the corresponding chimp chromosomes 2A and 2B remain separate. Now, here is a diagram depicting the fused human chromosome number 2. Now, note 2 things here. you see that there are 4 base pair sequences on each side of the exact fusion site, instead of the normal 6 base pair sequence. That's the first thing to notice. On the left side, there's the standard 6 base pair sequence. But on the right side of the fusion site, there's an abrupt change. There's an introduction of a new set of base pair repeats. And this can be explained in the following way. Now, here's a standard base pair sequence, just a single base pair sequence, right? Now, if you rotate this molecule or this set of molecules, if you rotate it anti-clockwise 180 degrees, it yields the following sequence. And this sequence is exactly the sequence found to the right side of the fusion site. So in other words, we now, with this background information, we can reconstruct the chromosomal history of our ancestors. We're going to focus on the 4 chromosomes involved in the fusion process. Remember, the 2 human chromosomes before fusion and the corresponding chimp chromosomes. Now, here's an illustration of the 2 human chromosomes before fusion. You should remember this happened hundreds of thousands of years ago. And what you should notice here is the 6 base pair sequence being repeated, which is found in the telomeres in the heads of the human chromosome. So what you have to note here is that the 2 ancestral human chromosomes before fusion are highly similar to the 2 corresponding chimps chromosomes 2A and 2B. On the sketch, it appears that it's identical, but actually there's some minor differences. But let's forget about the differences, because just to facilitate understanding, assume they are almost identical. So what happens now in the human chromosomes before fusion? The tips break off of the telomeres, right? And in the process, each of these chromosomes loses 2 base pair sequences, which is demonstrated here. You can see there the tips breaking off, and in the process, there's a loss of 2 base pairs on each of these chromosomes. So now the next step is that the one chromosome rotates 180 degrees relative to the other chromosome, and this rotation results in the inversion of the basic sequence, which explains why the sequence to the right side is exactly the reverse of the sequence on the left side. And the 2 chromosomes merge at the broken ends, and this is the molecular representation of how they fuse and what is the result. Here you have your 4 base pair sequences on each side of the fusion site instead of the normal 6 base pair sequence, and you have here the reverse sequence to the right side of the fusion site as compared to the left side. Now let me repeat the last 2 steps, which I think is the essence of the argument. Let's look at the 2 ancestral human chromosomes before fusion. And then you have your 2 corresponding chimp chromosomes, which is highly similar. And also just note that the telomeres are found at the ends of the chromosomes in both the human chromosomes and the chimp chromosomes. Now, when the tips break off and the one chromosome rotates 180 degrees relative to the other chromosome, the telomeres originally in the separate chromosomes, which were now at the ends, is now shifted and occupy now the middle part of the fused human chromosome. But in the chimp chromosomes, the telomeres retain their position at the ends of the chromosomes. So one should therefore expect the middle section of the fused human chromosome should match with the telomeres found at the heads of the 2 separate chimp chromosomes 2A and 2B, which is exactly the case. Now, the last piece of evidence which supports fusion concerns the centromeres. Now, you'll recall each chromosome has a centromere, and it has a specific base sequence, and it's identified by as the constricted region in the chromosome. Now, if fusion took place, if 2 ancestral chromosomes fused, one would expect that fused chromosome to have 2 centromeres. If you look at the fused human chromosome number 2, there seems to be only 1 centromere, and this centromere corresponds to the centromere on the chimp chromosome 2A. The other centromeres seem to be missing. But when scientists zoomed in on this human chromosome number 2 at the site where they expect to find the missing centromere, they found remnants typical of a centromere sequence. In other words, compelling evidence that this centromere once existed but has now been inactivated by mutational buildup. We see therefore that the above sequence of events and observations is beautifully explained by the findings, and that is, it explains why the middle section of fused human chromosome number 2 corresponds to the telomeres found in the heads of chimp chromosomes 2A and 2B. It explains why you have 4 bases on each side of the fusion site instead of the normal 6 base pair sequence. It explains why the sequence of bases on the right side of the fusion site is exactly the reverse of the bases on the left of the fusion site. And it explains why there are actually 2 centromeres as proposed by the fusion theory. All these findings can only be explained in terms of common ancestry. But this is just the tip of the iceberg. There is even more evidence. Now I just want to touch on transposable elements. Now if you sequence the DNA, you find that there are certain genetic sequences which seem to move around in the genome. So DNA is not a static molecule. It consists of segments which excise themselves and insert in a random fashion at other locations in the chromosomes. And these are called transposable elements. And a transposable element has a specific DNA sequence. Now, you'll agree with me that chances of a particular transposable element excising itself and inserting itself at an identical location in the DNA in 2 separate species is very small. But there are more than 1 million of these transposable elements in the chimps and humans. And you find that almost without fail that the same transposable element being located at the identical site in the chimp lineage and in the human lineage. Now the chances of this happening by accident is wildly improbable, and the most logical explanation that this common pattern was inherited from a common ancestor. Now the next piece of evidence concerns pseudogenes. Now as the name indicates, pseudogenes are genes which have lost their function because normally as a result of a change in behavior of the host animal. Now these pseudogenes are not immediately removed from the genetic material. They remain there for extended periods of time at the exact site where the once active gene was, and provide us with intriguing glimpses of our ancestral past. Now the most widely studied pseudogene is the vitamin C gene. Carnivorous mammals all have the active form of this gene, since they live almost exclusively on a meat diet devoid of vitamin C, which means that they have to manufacture their own vitamin C, which is a life-sustaining metabolite. All mammals possess this gene, except primates, which includes humans, guinea pigs, and fruit bats. Now, the reason for this is, remember, primates evolved a little bit later after the mammals. And these primates adopted a different lifestyle. They started to eat fruit, which contains large amounts of vitamin C. In other words, the vitamin C gene was no longer required. And when it was inactivated by a mutation, it didn't have any side effects on the animal because the primates obtained enough vitamin C from the diet. So why make a gene in any case, which is costly, when it's not needed? So what is striking is that you find that the mutation that disabled the vitamin C gene in primates is exactly the same chemical change in all primates. And the only reason for this is that this mutation happened in a common ancestor of the primates and was then transmitted to its descendants. We are also primates. We have the identical mutation as all the other primates. However, a different mutation inactivated the vitamin C gene in the guinea pig lineage. And the reason for this is that this mutation happened independently in the guinea pig line, which had already diverged from that of the primate lineage. We're going to revisit this idea of pseudogenes when we discuss the Adam and Eve creationist vision. But just for the moment, you have thousands of these pseudogenes in the chimpanzee and humans. In other words, useless genes. And you find the identical pseudogene at the exact location in the chimp lineage and in the human lineage. Again, when scientists sequence the chimp DNA and human DNA, repeatedly find the same sort of pattern. And the only, again, logical explanation is that this common pattern was inherited from a common ancestor. Now, the last concept I want to discuss or touch upon is that of chromosomal elements. When I said earlier on that we share 98% similarity with the chimps, it's a bit misleading in the sense that 9 or 10 of these elements, although they are essentially the same in chimps and humans, they occur at different location sites on the chromosomes. We said earlier that we emerged from a common ancestor. Now, for a species to give rise to two daughter species, what you need first is reproductive isolation. This can happen as a result of geographical factors such as a mountain range or a river or so, where some of the members of a particular species separated from the other species. So they can develop, then evolve independently, build up mutations, and become progressively genetically unlike. And this is what happened with the ancestors of the chimps and humans. So you have reproductive isolation, you have separation, you have build-up of mutational differences. If we believe that there's a common ancestor which split into two lineages, and these two species now cannot interbreed anymore, one should be able to find, one ought to find rather, chromosomal elements. And herein lies the main difference between chimps and humans, in chromosomal elements. So if two species emerge from a common ancestor and they diverge because of geographical and other factors, this allows them to evolve in separate directions and become genetically dissimilar progressively more and more. So if they should then come together again, they won't be able to interbreed because of these chromosomal elements. The main difference for the infertility is the chromosomal elements. If you examine two species which are closely related, which you propose that they emerge from a common ancestor, but they cannot interbreed anymore, you ought to find chromosomal elements. And this is the case with chimpanzee and humans, horses and donkeys and rats and mice.